Hi class. Please make sure you have your Chapter 7 note packet in front of you, a highlighter, and something to write with. We're going to do these notes similar to the way we did Section 1 and 2. You'll be highlighting things, adding things to the notes when I'm instructing you to, or trying to help you learn some of these note-taking skills and figuring out what's important and how to listen to what the instructor is saying to you. So this section is going to talk about cell boundaries, and specifically we're going to be looking at the cell membrane. And we'll be talking about how materials can move in and out of cells, because cells need things to survive. They need food and oxygen, and they need to get rid of wastes as well. So we're going to look at how these materials can get in and out of the cell. The key concept questions we'll be thinking about are: what are the main functions of the cell membrane and the cell wall? What happens during diffusion? And what is osmosis? So the cell membrane is composed of two phospholipid layers. The cell membrane has two major functions. It forms a boundary between the inside and the outside of the cell, and it controls the passage of materials. So the cell membrane is going to control what can enter and leave the cell. The cell membrane does not really give a rigid structure or support to a cell because it's a very flexible material. So please don't get it confused with the function of a cell wall, for example. The cell wall is mainly for support. I want you to add to this slide that cell membranes, they call cell membranes multiple different types of things. So sometimes they'll call a cell membrane the phospholipid bilayer. So you can add that to the slide. You can write that they also will call it a plasma membrane. These are all the same thing, but they'll use different terminology sometimes, and I don't want you to be confused. They're talking about the same thing, they just use different words for it. Here, you can see the cell membrane is made of a phospholipid bilayer. Please highlight phospholipid bilayer. And there are other molecules embedded in the membrane. It's not just all these phospholipids, there's other stuff in there. And because there's so many different things scattered throughout the membrane, they call it a fluid mosaic model to describe the way that the membrane looks. If you've ever seen a work of art that they call a mosaic, you can see how it has lots of different colors or textures to it. That's probably why they use this term, because the cell membrane has lots of different things in there. What I would like you to do is kind of give a short description of what some of these things do that are embedded in the membrane. So you can add these to the side of the picture, but we'll start with the proteins. Any of the proteins, they have different jobs to do within the membrane depending on what kind they are. So they could form a channel, they could work as a pump, or it can function as a receptor. And as we remember, the detail about proteins is they have a specific shape to do their particular job. The next thing we're going to look at are the carbohydrate chains. These chains act like chemical ID cards or name tags. It helps the organism identify what cell is which. It can identify that that cell belongs to that particular organism. So these carbohydrate chains work like name tags for the cells. Then there's cholesterol. They're embedded like within the tails, and these little cholesterol molecules help prevent the tails from sticking together, because sometimes they could if there's not enough of those cholesterol molecules there. The next slide will be a video clip discussing the membrane. You should keep this page in front of you, and you can add any labels that you may need to, or any extra descriptions, other words that they might use in the video clip, just to help you remember what these different things do.
The plasma membrane is composed of a bilayer formed by two chains of phospholipid molecules. The fatty acid tail of the phospholipid molecule is nonpolar, whereas the head of the phospholipid molecule containing the phosphate group is polar. The two phospholipid molecule chains layer like a sandwich, with the fatty acid tails forming the interior of the membrane and the phospholipid heads facing the watery environments found inside and outside the cell. Cholesterol is also found in the plasma membrane. It helps to stabilize the phospholipids by preventing the fatty acid tails from sticking together. Proteins that span the entire membrane form the selectively permeable membrane that regulates which molecules enter and which molecules leave a cell. Carbohydrates sticking out from the cell surface identify chemical signals and prevent the cell from succumbing to infection.
So the cell membrane has a quality that they use, that's term called selectively permeable. Please highlight that. And basically what it means is that some molecules can cross the membrane and others can't. Think about what it means to be selective. If you're selective, that means you're being picky or choosy. And permeable means it can go through. So if you think about perfume can permeate or move through a room, you can think about the word permeable that way. It allows things through, kind of like a window screen. I think I've heard someone talk about the cell membrane like a sieve, that you would strain food in. And just so you're aware, the outside of a cell has fluid in it, just like the inside of the cell does. So the membrane acts to control how much fluid can go in and out and all the other materials that are going in and out. The cell membrane, like we said, is selectively permeable, and you can see how some molecules can easily pass through here. The little blue ones can pass through. They're small. They don't have an electrical charge. They can just slip right through, like carbon dioxide and oxygen can do this. So you know what? Why don't you add that, that small molecules can pass through and non-charged or uncharged molecules can pass through easily. Chemical signals are transmitted across the membrane. Obviously, cells need to communicate with each other. If you think about the trillions of cells in our body, they have to have a way to talk to each other so that they know what each other is doing. And this is part of homeostasis. So receptors, we said proteins are types of receptors, or receptors are types of proteins, I should say. They will help this communication occur. And what I want you to do, where it says receptors here, before we get into the rest of the detail about them, I want you to add next to there that they are proteins, because then that hopefully helps you remember that they have to have a specific shape to do their job. So there's different types of receptors. They will bind with a ligand, and a ligand is just a molecule. It's a chemical that is sending a message. There can be intracellular receptors. That means there's a receptor on the inside of the cell, so that ligand or that message has to get through the membrane and find that receptor, bind to it, and then the cell goes, oh, okay, I know what you're doing, I know what you're saying. And then the other type of receptor is called a membrane receptor. And this is where the protein or that receptor is embedded in the membrane itself, and it receives that ligand or that message from outside. And as soon as it binds with that ligand, then it can release a message to the inside of the cell. Just so you know how these receptors work, there's a couple different ways, and these are two of the ways that these receptors work. Materials can move across membranes because of a concentration difference or concentration gradient. When the concentration of a solute is the same throughout a solution, the solution has reached equilibrium. And that's essentially the goal of all of the molecules that you ever talk about, is they want to reach equilibrium. Molecules will move until they're equal on both sides of the membrane. Now, that doesn't mean they stop moving. They'll still move, but they'll move at the same rate across the membrane. But like I said before, we've talked about things that the cells need, like nutrients and oxygen. They have to get rid of wastes. So the question is what makes these materials move across the membrane? And it has to do with the concentration differences in these molecules. If there's not a lot of oxygen inside the cell, oxygen is going to want to move in to balance that out. So that's the goal of the cell, is trying to get these things in there with using the least amount of energy. So one of the ways that molecules move is called passive transport. Passive, that means you're not doing anything. It means you're not using energy. Same thing with the cell. Passive transport does not require energy for the cell at all. So molecules can move across the cell membrane through passive transport, and there's two types that we'll talk about: diffusion and osmosis. And the little diagram there you saw with the purple square, or the purple circles inside the square, they moved through diffusion until they were equally spaced apart. In the right-hand picture, those molecules are going to want to move from the left side to the right side, or into the cell, to become equal on both sides, and that's through the process of passive transport. So diffusion and osmosis are types of passive transport. Molecules will diffuse down a concentration gradient. They want to go from where there's more to where there's less. It's kind of like riding a bike down a hill. You don't have to do anything. You can just sit there and the bike will move because it's going from a high energy to a low energy state. Well, these molecules are going to move where they're in a higher concentration to where they are in a lower concentration. I'm actually going to have you do what they're doing in this picture. You're going to be putting a drop of food coloring in a beaker, and we won't do anything to the beaker, but eventually all of those little color molecules will be equally spread out, and it won't require any energy to do that. So one of the definitions you really need to remember is diffusion. So put a star next to it, make sure you highlight the word diffusion. This is the movement of molecules from an area of high concentration to an area of low concentration, like going downhill. And then osmosis is very similar, but it's very specific because it's the diffusion of water molecules from where there is more water to where there is less water. And most of the time, osmosis is used in conjunction with a semipermeable membrane, because water doesn't just diffuse randomly through water. It has to go through a membrane. There has to be a difference in the water concentration. Okay, so let's start with a little bit of practice with diffusion. Very basic picture. The little black dots represent molecules, the white circle represents a cell, and then the gray background is the outside of the cell. So the question I'm asking is, how will these molecules diffuse? What will the final picture look like? And I want you to try and draw it before I show you the answer. So what you want to think about is, where are there more dots or more molecules, and where are there fewer molecules? Which way will these molecules want to move? Will they want to move into the cell, or will they want to move out of the cell? Remember, their goal is to be equal on both sides. So think about it, draw in what you think the final picture will look like, and then I will show you. So here we have our final answer. Did you get the same amount? It doesn't matter where you drew your dots, just that you drew the same number that I have inside and outside the cell. They want to reach equilibrium. So that's what this looks like, is this is equilibrium. Okay, now we're going to try this with water. We're going to talk about osmosis. So in this situation, you'll think about where is there more water, where is there less water? Which way will water want to move? Again, water wants to diffuse until it reaches equilibrium, until it's the same inside and outside the cell. So figure out which percentages will end up where, and then I'll show you the answer. Did you come up with 50% inside and outside? And you can even draw an arrow on your picture showing that water wanted to diffuse into the cell. Okay, so this slide is, it's got a lot of information on it. We're going to spend a minute or two talking about these three terms, isotonic, hypertonic, and hypotonic. And we're going to use these words to describe the conditions inside and outside cells. And these are comparative terms, so you're always using them and when you're comparing two solutions to each other. On this slide, I want you to add that most cells have, as far as concentration goes, about 1% salt and 99% water in them. Okay, we're going to use that as a baseline for any cell that we talk about, just so that you're aware. If you've ever been in the hospital and you've had to have fluids put into your vein, they give you a 0.9% saline. It's almost 1% salt. And that's because they're trying to be very close to the salt concentration that your cells have. They don't want your cells doing funky things because you're obviously sick if you need to have IV fluids. So next time, hopefully it won't be anytime soon, but if you ever need fluids again, take a look at the bottle and see if you can find that they are giving you a 0.9% saline. Sometimes they give you another one that's called lactated ringers, but if you look carefully at it, you'll see the concentration is pretty close to 0.9%. Okay, so now these three terms. Knowing the difference between them. Isotonic, and this is the first diagram there, you're actually seeing a picture of a red blood cell. So if a solution is isotonic, you're comparing that to the inside of the cell. Isotonic means the same. So next to the term isotonic, you're going to write same. The concentration inside and outside the cell in an isotonic solution is going to be the same. So it might be a salty solution, 0.9%. That's like the saline you get in the hospital. That's an isotonic solution. It's the same concentration as it is inside your cell. Hypertonic solution. Don't get it confused with hypotonic. Okay? The way I remember the difference between these, hypertonic solution is going to be more concentrated in a solute or salt. So if I think about hyper, if someone is hyper, they move around a lot. So take that a lot, you know, that term a lot, they move around a lot, and you can write it next to hyper. There's a lot of solute or salt. You can add that to your notes. And then hypotonic, hypo and no or low, they will have no salt or low salt amount. So hypotonic will have less salt concentration than the inside of a cell. So you can see what happens to a cell in these pictures. If you take a cell that just has a cell membrane like a red blood cell and you put it in an isotonic solution, nothing really happens to it. It looks the same. Water will move in and out at equal amounts. It'll be at equilibrium already. If you put a red blood cell in a hypertonic solution, it's going to shrivel. Sometimes it could die, depending on how hypertonic that solution is. And it does this because the concentration of water is actually higher inside that cell than it is outside. And water is going to leave the cell so that it can make it equal. Then in a hypotonic solution, this is like, let's say pure water, for example, the water is actually going to be more concentrated outside and it's going to diffuse into the cell. And if too much diffuses in, it could expand the cell and then make it burst. And that would be bad. So that's why they don't give you pure water in the hospital, because your cells would all just explode. And then you wouldn't be able to, you would die. Like if you know what red blood cells do, they transport oxygen through your body. So if they all burst, there aren't any cells to move oxygen around, and that's a big problem. So I would recommend that you pause the video and listen to this part again so that you can get hypertonic, hypotonic, and isotonic straight. You want to know what these different terms are talking about. Here's another diagram just talking about the same terms we were just talking about. The arrows are showing the movement of water. So I want you to add that. So the hypotonic picture, I want you to add next to one of the arrows that water is moving in. The hypertonic, next to an arrow, you can write water is moving out. And then isotonic, you can say water moves in and out at the same rate, because it's going to be the same concentration. It's already at equilibrium. And here we have, again, another diagram showing you what happens to cells when they're placed in the different types of water. Well, I'm going to quiz you a little bit or challenge you a little. I want you to write down the term hypotonic, hypertonic, and isotonic above where they belong with, is it freshwater, balanced, or saltwater? I want you to write them where they belong as far as these types of solutions. So pause the video, write down which one you think is hypertonic, which one is hypotonic, which one is isotonic, and then I'll tell you what the correct answer is. Okay, so freshwater. You can see that the cell is bursting or lysing. This is going to be hypotonic. This is like pure water, okay? Balanced, that's going to be your isotonic because it's at equilibrium. Water goes in and out at the same rate. And then the salt water will be your hypertonic solution. And you can see animal cells in freshwater will burst because they don't have anything, the cell membrane is not strong enough to withstand that much water. For a plant cell at the bottom, it's going to become turgid, and that's actually what plant cells want. So when you water a plant, their vacuole, their big water vacuole, fills up with water and puts pressure against the cell wall, and it causes the plant to be able to stand upright and look pretty happy. Then if you go away on vacation, the plant cell does what the middle picture is doing. It becomes flaccid, and that's where you come back and the plant's kind of droopy, not looking very good. And that's because it's sort of at the balanced state, and that's not very good for a plant cell. However, in an animal cell, that is a better state for that cell. Then if you were to pour salt water on any kind of cell, water is going to leave the cell and cause them to shrivel. In an animal cell or a plant cell, does something called it plasmolyzes. We're actually going to do this to plant cells. We're going to take some red onion cells and put some salt water on them, and you'll actually see the cell membrane pull away from the cell wall and kind of shrivel up inside. It's actually kind of neat when it does that.
During osmosis, water diffuses across the cell's selectively permeable plasma membrane. Regulating the flow of water through the plasma membrane is an important factor in maintaining homeostasis of the cell. Let's look at 3 examples. In an isotonic solution, the concentration of dissolved substances in the solution is the same as the concentration of dissolved substances inside the cell. Likewise, the concentration of water in the solution outside the cell is the same as the concentration of water inside the cell. The diffusion of water into and out of a cell occurs at an equal rate. The cell retains its normal shape and pressure. In a hypotonic solution, the concentration of dissolved substances is higher inside the cell than the concentration of dissolved substances outside the cell. Water passes through the cell's plasma membrane into the cell, causing the cell to swell. In plant cells, the plasma membrane swells and presses against the fairly rigid cell wall, preventing the cell from bursting. In a hypertonic solution, the concentration of dissolved substances is lower inside the cell than the concentration of dissolved substances outside the cell. Water passes through the cell's plasma membrane and exits the cell, causing the cell to shrink. In plant cells, the cell's plasma membrane shrinks and pulls away from the cell wall.
Okay, some molecules can only diffuse through transport proteins. So far we've been talking about passive transport. These are small molecules that have no charge, and they can pass through the membrane without any help. Some molecules need a little bit of help. They don't easily diffuse through the cell membrane, and this is called facilitated diffusion. If you facilitate, you're helping. So there's a protein involved that helps these molecules pass through the membrane. Please make sure you highlight facilitated diffusion. And next to facilitate, I want you to write the word help. These transport proteins are helping, okay? And then another type of protein is going to help a little bit more, but it needs energy to do it. And there's three types, or actually there's probably more than that, but we're going to talk about active transport, endocytosis, and exocytosis. All of these processes require energy to move molecules across the membrane. Remember your prefixes and suffixes. Endo means into. So endo means things are coming into the cell. And then exocytosis means things are leaving the cell. If you think of exit and exo, they both have the EX. Things are exiting the cell for exocytosis. Why don't you write that little hint down next to exocytosis? You could write exit or leaves the cell. Endo means going into the cell. Okay, so talking about active transport, it's going to require energy. And if you think about the term active, it requires energy for you to be active. So that might help you remember active transport needs energy, because usually what's happening is that the cell is trying to move molecules against the concentration gradient. If you think about when I said before, passive transport is like riding a bike down a hill. You don't need energy to do that. Active transport is the opposite of that. It's you pushing that bike up the hill. It takes a lot of energy to get that bike back up the hill. So passive transport, no energy. Active transport needs energy. The type of cell energy we're talking about is short, it's 3 letters long. It's called ATP. So what I want you to do next to that bullet where it says ATP, I want you to write cell energy. You're going to see ATP come up a lot this year. It is short for adenosine triphosphate. You don't have to write that down, but just so you know, that's what ATP stands for. It stands for adenosine triphosphate. So active transport occurs through transport protein pumps. They're actual pumps that have to move things against the concentration gradient. And they have to use active transport to maintain homeostasis. Cells have to do this for certain things. Okay, so a cell can import and export large materials or large amounts of material in vesicles during the processes of endocytosis and exocytosis. So cells use energy to transport the material in the vesicles, and vesicles are just like little bubbles of stuff. Please highlight endocytosis. This is where the cell is bringing material into the cell. Phagocytosis is a type of endocytosis, and the prefix phago means to eat. So this is kind of like cell eating process, or the cell is taking in solid material. I do want you to add that next to phagocytosis, and that means the cell is bringing in solid materials. Then we have pinocytosis. This is the cell taking in liquids from the environment. So it's almost like the cell drinking, sort of. So next to pinocytosis, I want you to write down that it's bringing liquids in. Okay, so exocytosis. This is the process of expelling material from the cell. It's kind of getting rid of waste. It's getting rid of large amounts of materials.
The substances used as fuel by single-celled organisms include other smaller cells, particles of organic material, and large molecules that cannot pass through the plasma membrane. Many single-celled eukaryotes use a mechanism called endocytosis to ingest such food particles. In this process, the plasma membrane surrounds and engulfs the food particle. Cells use three basic types of endocytosis depending on the size and nature of the material to be ingested: phagocytosis, pinocytosis, and receptor-mediated endocytosis. If the material the cell takes in is particulate, such as a bacterial cell or an organic fragment, the process is called phagocytosis. If the material is a liquid, it is called pinocytosis. Some types of molecules, such as low-density lipoproteins, LDLs, are transported across the plasma membrane by receptor-mediated endocytosis. These molecules first bind to specific receptors embedded in the plasma membrane. The receptor molecules are concentrated in an indented pit coated by the protein clathrin. When sufficient target molecules accumulate in the coated pit, the pit deepens, seals, and is incorporated into the cell as a coated vesicle. Exocytosis is the reverse of endocytosis. This process results in the discharge of materials from membrane-bound packages that migrate to the inner surface of the plasma membrane, fuse with the membrane, and then release their contents to the outside of the cell.
Okay, so there are some unicellular, one-celled eukaryotes, so they have membrane-bound organelles and a nucleus, and they have this cool structure called a contractile vacuole. And what it does for these guys is it'll collect excess water that's inside the cell and force it out of the cell. If they didn't have this, it's like a pump, like a little water pump, these contractile vacuoles. And many of these organisms live in fresh water, so very high water concentration compared to the inside of them. And if they didn't have this pump, they would burst, they would die. So they have this little pump that actually pushes extra water back out so that they don't explode. And I'm going to show you this link and show you what these cells actually look like. And you can see the little contractile vacuoles pumping the water as they're moving around. It's pretty neat. Okay, so I'll just talk you through this. It's just showing movement of particles moving through the membrane, just through diffusion. This diagram is going to show you just another simple diffusion. You've got a beaker of water, you drop some sort of a tablet and it dissolves, and then eventually the particles will spread out throughout the liquid and they'll reach equilibrium. All those little molecules are evenly spaced apart. Here we have the same beaker. You've got different concentration of solute on either side, and this is a selectively permeable membrane. If you let this sit, what happens is the water will diffuse to the right side to balance out the concentration of the molecules, and the water level will actually rise on that half. This showing you inside your body, you've got certain concentration inside your blood vessels. There's a hydrostatic pressure of water. Some of the molecules will essentially be forced out because they're in higher concentration. Scary needle, syringe. And then just showing you kind of like a hydrostatic pressure, and now if there's holes, you're going to see things coming out those holes and not out the end of the needle. And that's essentially the way the membrane is if there's enough pressure put on a blood vessel, for example, the excess will be pushed out through the holes in the membrane.